Okay, then. In this presentation, I'm going over this paper recently submitted, and it's under review. The name is TOMEXO, and it stands for Tree of Mutually Exclusive Oncogenes. And it is supposed to simultaneously infer mutual exclusivity and progression patterns among oncogenes. We go like this. Okay, so the cancer progression models are typically used for studying somatic mutations in cohorts of tumors, and they can be used as tools to identify cancer driver genes in abundance of passenger mutations and find interrelations among oncogenes, patterns such as mutual exclusivity, where mutation in some oncogenes exhausts the selective advantage of mutation in other oncogenes, or progression patterns where mutation in specific genes paves the way for mutation in other genes or increases the probability of mutation in other genes. And there are a lot of models doing more or less similar things, and these two path Timex and mutual hazard networks are two examples, and this is like the structures that they have. So these progression models typically put genes in a network or like a structure like that. So in this paper, we introduce a progression model, and our progression model includes the driver tree and a couple of error probabilities. The driver tree is something like this, and I will explain it more. The data that we work with is a binary matrix with the rows including the tumors and the columns being the genes. And basically, it is showing presence or absence of mutations in a set of genes in a cohort of tumors. And this driver tree can be seen as a generative model for generating tumor vectors. And the idea is that the driver tree in its node has a single oncogene or a set of mutually exclusive oncogenes. And on the edges, we have a firing probability for each edge. And here's how it generates the data. Starting from the root node, each edge has a chance of firing. Like here in this case, for example, both outgoing edges have fired. If the edge fires, then the node that it points to gets a mutation. And two things happen when a node gets a mutation. First, one and only one of its genes, uniformly selected, gets a driver mutation, and then each outgoing edge has a chance of firing according to its firing probability. Like here, for example, gene B has got a mutation, and then we have this edge has fired, and here gene E has also got a mutation. So this generative model gives a list of driver mutations, and then we have two types of false positive and false negative errors, which add on top of that and build our observed matrix. So in this paper, we introduce an efficient likelihood calculation to calculate likelihood of the whole data set, given each driver tree structure and the error parameters, of course. And we use it inside the MCMC inference algorithm. And our MCMC inference algorithm has a wide set of different types of moves, including both structural moves, different types of structural moves, and gene replacement moves. The structural moves include, for example, if this figure A is the current state, we may take a leaf node, like G, for example, here, and merge it into the parent, which leads to something like this. Or we may pick two leaves and merge them together. Here, we can pick this and this one and merge them together and get here. And we also have normal tree moves like SPR moves, subtree pruning, and regrafting moves. And we have some types of gene replacement moves like gene swap, where a leaf swaps its genes with a parent, or passenger to driver gene move like F, where we select a subset of genes from the set of passengers, set of genes not in the driver tree, and put it in one of the nodes of the tree. So with this MCMC inference algorithm, we infer the progression model and the error parameters for each data set. And we use some synthetic data experiments to show that our inference algorithm actually works when the data is generated using our generative model. We have 3 scenarios: a linear generative model, or a tree-like generative model, or a mixture of tree-like generative model. And we cover different error probabilities from 0.001 to 0.1, and different number of tumors in the data set from 50 to 500 to check in which setting we can construct the generative model. We define some metrics to evaluate the output of the inference algorithm and compare it to the generative model.
Sorry, can I ask a very basic question?
Sure.
It would be helpful to understand this. So you say this is your tree, is your generative model, but behind what is this generative model? So what is it generating for? Is it generating mutation for like one across the genes inside of one tumor, or?
Yes. So each tumor can, in this setting, each tumor can be seen as a list, as a vector, a list of mutations, and this tree plus the error events give a list of mutations.
In that tumor, and then
Yeah.
now this is generative model behind, you think, every tumor in.
Yeah, every tumor of like the same cancer type. Yeah, like we have say 300 tumors of a cancer type, and we assume that a single generative model is constructing all of these binary vectors. So each time.
And why is it a tree now? I mean, because so is there any interpretation that there is a node above, or is a predecessor of another node? So this means what?
So this means that. So what this means actually is that, for example, for a mutation in E, F, G, or H to happen, we have to have a mutation in A, B, C, or D. So it says that it can be seen like this. The cancer starts from no mutation. Then it may get a mutation in this set. And if it happens, then it may progress along this edge and get a mutation here.
And these probabilities are conditional probabilities given?
Yeah, these are the probability of, like, for example, here 0.7 is probability of getting a mutation in this set if we have a mutation in this set. Yeah. Okay, so and then each one of these structures imply a set of mutual exclusivity and progression relations. For example, here we have A, B, C, D in a set. And it means that these genes are mutually exclusive. So we can say, for example, that A and B are mutually exclusive, A and C are mutually exclusive, and so on. So it gives a set of mutual exclusivity relations and also a set of progression relations. For example, here it's saying that A is getting a mutation before E, for example, and A is getting a mutation before I. And then we check to what extent we recover the exact same relations in the model that we are outputting in our inference. So basically, these scores are, when the score is 1, it means that the inference algorithm has outputted the exact generative model. And we see that in cases where the error is up to 0.05, especially if the number of tumors is above 100, we can construct a very similar, and the output is very similar to generative model. So the inference algorithm is kind of working in all these three scenarios. Yeah, and then we study several biological data sets. This is glioblastoma multiforme, where we have 290 tumors, 16 genes in the data set. And this is the complete data set, binary data set that we work with. And this is the progression model that we construct. And then we define some metrics for this mutual exclusivity and progression relations, which are implied by our model. And these metrics are method independent. For example, here we have this inside each node where it's claimed that these two genes, for example, are mutually exclusive. We check the mutual exclusivity among these two genes in the data set. And here, mutual exclusivity score of 0.27 means that probability of mutation in PIK3R1, for example, is multiplied by 0.27 if we have a mutation in PIK3CA. So mutation in PIK3CA reduces chance of mutation in PIK3R1. And we have a similar metric for evaluating these progression relations. And we also check the p-values of such things happening by chance, and we see that a lot of these signals actually are not that strong, and this is because of the small number of tumors. The signals cannot be super strong, but there are several interesting signals. For example, here, TP53 before IDH1, we see that the p-value is very good, and in this data set, actually, there is not a single tumor with IDH1 unless we have also a mutation in TP53. And there are a lot of, so IDH1 mutation rate is not that low, for example. So some of the signals are strong, like TP53 to RB1 or TP53 to IDH1, IDH1 to ATRX, and so on. Okay, yeah, I'm already almost done. And then we also study some smaller data sets like this one, cholangiocarcinoma. And in this case, of course, because we have only 35 tumors and 14 genes, the signals cannot be really strong. And you cannot actually, if you just check the pairs of genes, you cannot find any strong mutual exclusivity or progression signals. But our likelihood function still gives us something like this. And yeah, so we have big sets of mutual exclusive genes. And actually here, we didn't need any error events to generate this. So this progression model without any error events can construct a complete data set. And so to sum up, we introduce a probabilistic model for cancer progression and an efficient likelihood calculation algorithm. We designed a set of novel MCMC moves for the specific structure that we have, which includes both structural moves and gene replacement. And our per iteration computational complexity is linear in number of tumors and number of genes, which is due to our efficient likelihood calculation and enables us to work with big data sets. Our biological data analysis recovers several well-known relations between oncogenes and suggests some interesting new ones. And this method actually extends our previously published model and linear progression models. In so many ways, the structure is extended from a linear structure to a tree structure, and because of our moves, we have automatic model selection, which we didn't have in our linear progression model. And our computational complexity has also improved a lot because of an efficient error estimation module that we have inside the MCMC. And that's all. Thank you for your attention. Are we on time? Yes?
So the input for this method, do you use only mutations or also copy number alterations?
In theory, it can be any event, but we work with just somatic mutations. But there are progression models which take also copy number variations similar to.
So you're interested into mutual exclusivity between amongst oncogenes, but then for both cholangiocarcinoma and glioblastoma in the list of genes, I don't know how you selected the genes, but you had like PTEN, P53, CDKN2A,
Yeah.
which are well-known tumor suppressors. So are you...
Yeah, so the list of genes to include are selected based on, they are potentially driver genes, like the genes which have the potential to be in the driver tree, and they are selected based on their mutation frequency in the data has to be higher than a threshold, and they also have to be included in this Intogen list of potential. Intogen is a project done by Nuria Group, and it provides a list of drivers for each cancer type. So we intersect that set and the set of genes which are mutated with the frequency of mutation higher than a specific threshold. So those are.
And you kept both oncogenes and tumor suppressors?
Yeah, yeah. Mm-hmm. Yes? Okay. You.
Because it was simply because you seem to apply like a timeline of the acquisition of these events through this model, but the timeline is exclusively based on the occurrence of the alteration in the
Yeah.
data set, right? Just want to make sure I
It doesn't explicitly suggest time. So time is not involved in this model, but it can be seen as like TP53 has to happen before IDH1 if we have tumors with TP53 and tumors with TP53 and IDH1, and no tumor with IDH1 and no TP53. So.
okay, so it's based on the occurrence.
Yeah, yeah.
When you say before, I mean you're implying a timeline.
No. Yeah. No, but this is just introducing a distribution over the set of observed mutations, effectively. Or we can talk about it more.
Take one question, and I think.
One more question. Okay. Who? Hania was trying to ask a question for long.
So I just wanted to ask, here, do you encode just the 0-1 status? So, for example, that TP53 is mutated or not mutated?
Yeah.
Yeah.
But
do you take into account any mutation? So can it be synonymous or non-synonymous mutations? Or do you somehow...
No, I filter the mutation and consider those which change the, I don't know the exact biological name, but change the codon or has some functionality. Yeah.
And do you think about taking into account, for example, also lists of mutations that are known to be driver mutations or passenger mutations in driver genes? Because this could also...
For each gene, yeah, we have a list of mutations. That can be, yeah, that should be beneficial to use that. Yeah. Yes?
Maybe we can take one more question.
Yes. Okay.
Sorry. Just a quick question. So are all the mutations you're looking at pre-treatment or some of them are post-treatment? Because you might imagine that treatment might actually
I think
impact the mutations that are observed. And the second case is to differentiate between activation and inactivation mutations.
yeah. So this is based on TCGA data. I assume that there are pre-treatment, right? I'm not really sure that it's okay. Or are they mixed? Yeah. Okay. So I don't know about that. And what was the second question?
To differentiate between activation or inactivation mutations.
I don't know what activation mutation mean actually. Yes. Sorry for that. Yeah. Okay, can we have a question or should I?
I think maybe we can raise it.
Okay, sorry.
Yeah, thank you.
Thanks.